Hey everyone, my name is Chengyu, and I'm a postdoc in the HIT lab at the UCSF. So today is a great opportunity to share with you our experiences with massive parallel reporter assay and how to use this technique to decode the regulatory effects of genetic variants. So before I dive into the technical details, I'd like to briefly introduce the HIT lab. So this is the picture of my mentor Nadav and the HIT lab. So our lab have spent extensive efforts in developing MPRA protocol as well as building computational analysis pipeline and research sharing platforms. And our lab has done tons of MPRA to study genetic variation in human disease, evolution, and then development. Okay, so just a little bit backgrounds. One of the biggest challenges in population genetics is how to identify the causal variants out of a vast number of hits. And this is a crucial step to understand how genetic variation lead to phenotypic changes. And the challenge lies in the fact that most of the variants are located in non-coding region with unknown function. And we have very limited knowledge about the grammars in the non-coding region. So often we don't know the function of a particular non-coding region. And even when we do, we may still confused about how genetic mutation could affect its function. And many of the non-coding regions actually regulate gene expression. So they are gene regulatory elements such as enhancers, promoters, or silencers, and they either activate or repress gene expression. And genetic mutations within these regulatory elements could affect their activity. And the challenging is that the gene regulatory activity is very cell type specific, and some of the elements may only active in a certain developmental points or under certain conditions. So this cell type and spatial temporal specificity adding another layer of complexity, making it very challenging to understand the function of the non-coding region and how genetic variation affect the regulatory function. And to address all these challenges, MPRA can be a helpful tool. So in my talk, I'll briefly introduce the technique, and I'll give two examples of how this technique has been used to understand the genetic variation. Then I'll provide guidance of how to design an MPRA library, how to carry out experiments and the data analysis. And lastly, I will mention the current limitation of the techniques. So before I introduce MPRA, I'd like to briefly introduce what is a reporter assay. So imagine we have a region of interest, and we want to find out whether this region functions as an enhancer or promoter that activate gene expression. And we can do this using reporter assay. So the idea is to clone the sequence of interest into a reporter construct in front of a reporter gene. In this example, we have luciferase genes, which encodes a luminescence protein. So if the test sequence functions as an enhancer or promoter, it will drive the expression of the luciferase genes and leading to the luminescent intensity. So we then measure the amount of luminescence we observe, and this will quantify the regulatory potential of the test sequence. Now, let's say if we have a variant of interest, let's say it's an A2G mutation, then we can clone two reporter vectors. One contains the sequence with the A allele and the other with the G allele. And then we measure the luminescent intensity from the two vectors and compare them. So if there is significant difference between the two, then we can see that this A2G mutation indeed has a great impact on the gene regulatory activity. Well, this reporter assay is quite informative, but it has a major drawback, and that is we need to run each sequence one by one. So if you have a lot of sequence we want to test, a lot of variants we want to test, and running this reporter assay one by one will be very low efficient. So one way to address this low efficiency is through massively parallel reporter assay, or MPRA. So, and the key for increasing throughput is the use of barcode. So shown here is a standard reporter vector. We have our candidate enhancer and a minimal promoter and a reporter gene. And then we tag a barcode to the construct. So a barcode is a short and unique DNA sequence, usually 10 to 15 base pair long. It serves as an identifier for the sequence we want to test. And then we can test a lot of different candidate enhancers in parallel. Each one will be tagged with a unique barcode. And then we measure the expression of the barcode through high-throughput sequencing. And the barcode expression level will represent the regulatory activity from the test sequence. So the idea is using barcodes so we can measure a lot of sequence at once. And another version of MPRA is called StarSeq. So the major difference is in a StarSeq, instead of using a barcode, it used the enhancer itself as a barcode. So the enhancer sequence is now put downstream of the reporter gene and replace the barcode sequence. The idea is that the enhancer can function regardless of orientation. So if the test sequence is enhancer, it will drive the expression of the reporter gene as well as drive the expression of itself. But the way the StarSeq construct could introduce some bias, like the RNA polymerase could interfere with the transcription factor binding in the enhancer sequence. So for the rest of my talk, I'll focus on MPRA. So now I show one example of MPRA-based saturation mutagenesis. So this is one study from our lab a few years ago. So we introduced all possible mutations in each position of many disease-relevant promoters. Third promoter shown here as an example. So in this plot, the x-axis is the genomic location of the promoter, and the y-axis represents the change in enhanced activity measured by MPRA. So we measure the enhanced activity of a lot of mutated sequence and then compare it with the wild-type sequence. So the results indicate that the mutation effects is highly dependent on the location as well as the type of mutation. So G2A mutation in the middle of the promoter lead to the strongest impact on the promoter activity. And then the data of this MPRA is available through the MAVE database. Other than saturation mutagenesis, MPRA can also be used to identify causal variants among hundreds of thousands of disease-associated GWAS or QTL hits. So in a recent collaborative project between our lab and Katie Pollard's lab at UCSF, we tested over 17,000 SNPs associated with many psychiatric disorders. So most of the variant we test is associated with either schizophrenia or autism spectrum disorder, and then the rest are associated with other psychiatric disorder. And the MPRA was performed in primary cortical cell as well as iPSC-derived cerebral organoids. So we measured the enhancer activity of each sequence containing either the alternative allele or the reference allele of each variant. And then we compared activity between two alleles. So the result is summarized in this volcano plot. The x-axis is the fold change in activity between two alleles, and y-axis is the log p-value. So we found that most of the variants we tested had mild effects on enhanced activity, and we only identified 164 variants with significant allelic difference in their regulatory activity. So this result indicates that many of the variants identified from an association study may be neutral with minimal effects on gene regulation. So this highlights the importance of running high-throughput screening like MPRA to identify the small subset of functional variants. So the first step to perform MPRA is to design a library. And then we can select the variant of interest from any sources, can be either from the GWAS, QTL, fine mapping, or saturation mutagenesis. And we can then further filter the list of variants we want to test. So based on our experiences, a practical number of variants to test in a single experiment is less than 100,000. Beyond that number, the library becomes too complex, and it's going to be challenging to obtain high-quality results. So we can further filter the variants based on the epigenomic annotation. We can select variants located in ataxic peaks or histomark gypsic peaks, or variants located in a chromatin contact loops. Regions with this annotation are more likely to have regulatory function. So if we focus on variants in this annotated region, we will have a higher chance to identify regulatory variants. And one thing to note is that this epigenomic annotation is quite cell-type specific. So it's important to use the right annotation for your target cell type. And then once we have a final list of variants, we will generate sequence usually centered around the variants. And the length of the sequence is between 200 to 900 base pair. Longer sequence give you more genomic context, but can also be more costly to synthesize. And then we want to include sequence for all the alleles that we want to study for each variant, like shown here. And another important thing we want to include in the library is control sequence. So we want to include positive and negative controls. These are sequences that are known to have high or low regulatory activities. Ideally, this sequence should be validated in a previous MPRA. And another type of control is scrambled sequence. So we randomly select a few hundred sequences from the library and shuffle the sequence. This will solve another type of negative control. And an important thing for the positive and negative control is that it varies largely between cell types. So a sequence can be a positive control for cell type A, but become negative control for cell type B. So it's very important to select control based on your cell type. And one way to do that is to search in previous MPRA data sets. So you can use a database developed in our lab. It's called MPRA base. So basically, it integrates a lot of public MPRA data sets. So you can search for MPRA data performed in your target cells to get positive and negative controls. And this database can be also helpful if you are interested in a particular region and want to know whether this region have enhancer functions in your target cell type. Okay, so the next step is carry out the experiments. So you can find a detailed step-by-step protocol from a previous article in our lab. And in this article, it also introduced MPRA flow, which is a computational pipeline to analyze the data, which I'll discuss it later. So shown here is the overview of the experimental procedure of a lentivirus-based MPRA. It's actually quite similar to the base editing screen just introduced. So on the bottom, it shows the estimate times for each step. So the first step is to synthesize the designed library into a pool of oligos. We can also call them candidate regulatory sequence, or CRS. And then we tag barcodes and a minimal promoter to each sequence through PCR. And then we clone this full-length sequence into a reporter backbone vector. So this will generate a plasmid library. And then we sequence the plasmid library to determine which barcode is tagged to which oligo or sequence. And the next step is to package the plasmid library into lentivirus. And one important question to determine before running the experiments is whether your target cell type can be infected with lentivirus effectively. So lenti usually has a broad trapezoid and can infect a lot of hard-to-transfect cells. But some cells, like iPSC or immune cells, they are more resistant to lentiviral infection. So after packaging lentivirus, titrate it and infect the target cells. We'll then extract DNA and RNA from the infected cells and prepare a sequencing library. So eventually, we want to use sequencing to determine the expression level of each barcode by calculating the RNA barcode abundance and normalize it with the DNA copy number. And this barcode expression will represent the regulatory activity of the test sequence. So this is just a workflow of lenti-MPRA, and there are other versions of MPRA with slightly different protocol, but the main idea is very similar to what I'm showing here. Now I'd like to mention the calculation in the lenti-MPRA experiments. So the first number to calculate is library complexity. So it represents how many unique CRS and barcode pairs in the library. And it is calculated by multiplying the number of CRS with the number of barcodes per CRS. So for example, we design a library containing 100,000 sequences, and then we decide to tag each sequence with 50 unique barcodes. This will give us a library complexity of 5 million. So there are 5 million unique barcodes. And another important number to determine is MOI. As introduced before, MOI represents the number of lentiviral integration activity per genome. And the maximum MOI depends on the cell type. So highly permissive cells would allow an MOI up to a few hundred, while hard-to-infect cells may only allow an MOI of less than 10 or 20. And the number of cells required for each replicase is calculated by multiplying the library complexity with the number of integration per barcode divided by the MOI. So because lentivirus integrate the reporter construct randomly into the host genome, and this could affect the results. So we want to integrate each construct into multiple different locations. Usually we're aiming for 15 to 500 integration per construct. So in this way, we will average out the results. And this also means that if your target cell type cannot allow a high MOI, you may consider reducing the library complexity by reducing the number of sequences in your library. And another consideration for the experiments is whether to do an episomal or chromosomal MPRA. So each method has its pros and cons, and the choice depends on the context of your study. So for episomal MPRA, it's usually delivered via plasmid transfection. So the advantage is the procedure is quite simple, rapid, and efficient. And also, episomal MPRA usually allows detection of broader range of activity. So shown below is a comparison between episomal and chromosomal MPRA testing the same set of sequence. And the y-axis shows the MPRA ratio or the enhanced activity. So you can see that in episomal MPRA, we see a huge difference in the enhanced activity between the positive sequence and negative sequence. Whereas in the chromosomal MPRA, the difference in activity is relatively small. Well, the downsides of episomal MPRA is that the reporter expression is transient. So it's not suitable to study gene regulation over a long time course, for example, during differentiation or reprogramming. And on the other hand, for chromosomal MPRA, like the one usually delivered via lentivirus, the reporter construct is integrated to the host genome, so it allows a relatively stable expression. And more importantly, it provides chromosomal context. This can be particularly important if you want to study the effects of chromatin. For example, if you want to study pioneer factors, which are known to affect chromatin accessibility, then doing a chromosomal MPRA is very necessary. And the downside is the increased technical complexity due to the use of lentivirus. And also I want to know that using plasmid transfection and lentivirus are just two commonly used ways to deliver the MPRA library. But there are other delivery methods, such as using AAV to deliver an episomal MPRA, or use the piggyback system to perform the chromosomal MPRA. And then the choice eventually, again, depends on the context of your study. Okay, so for the data analysis, analysis of MPRA data can be performed either using a custom script or using a computational pipeline. And I'll introduce MPRA Flow, which was developed in our lab. This is a user-friendly software, and the entire analysis will be completed with 2 commands in a terminal. So the input data will be the sequencing FASTQ file and a library design FASTA file, and a label file is optional. So the MPRA flow essentially carries out 2 major tasks. The first is association. The second is counting. So for the association step, it identifies which barcode is attached to which oligo. So it built a map linking each barcode to each sequence. And then in the counting step, so the pipeline normalizes the sequencing rates and count how many unique RNA molecule and DNA molecule for each barcode. And this will calculate an MPRA ratio. And this ratio represents the expression level of each barcode, and it also represents the regulatory potential of the sequence. So after analyzing the MPRA data, we usually generate a table of results like this. So each row corresponds to a sequence tested in MPRA, and we can name the sequence in whichever way we like, but usually we include information about the sequence in the name. So if the sequence contains a variant, we usually include the variant ID, whether the sequence contains a reference or alternative allele, the base of the allele, and the genomic coordinates. And then the next 5 columns are 5 replicates. So we usually run 3 replicates, but it's always better to have more replicates because it gives us more power to detect subtle effects. So the value in each cell is MPRA ratio. So a higher ratio means the sequence have stronger enhanced activity. And then we calculate the average ratio across replicates. And if the ratio is high enough across all replicates, we then call this sequence as active enhancers. And to determine the allelic effects, we eventually compare the MPRA ratio of the alternative allele with the reference allele. And we calculate a log-fold change. This will indicate the effect size and direction of the allelic effects. And I will also calculate p-value to reflect the statistical significance. And this is usually adjusted by multiple hypothesis testing. So there are multiple tools to calculate the log-fold change and p-value. And shown here is just a list of them. So we can either use the differential analysis tool that commonly used in RNA-seq to run the assay, like a DSeq tool. Or we can use statistical framework that is designed especially for MPRA data sets. And the results from different tools should be largely comparable. OK, so lastly, just like any other techniques, MPRA also has these caveats. And one important thing to note is that MPRA tests gene regulatory sequence within an artificial reporter construct instead of inside its native genomic context. And this might include some false positives. Another potential bias is that the sequence is tested with a minimal promoter, not with their cognate promoters. And one last thing is that MPRA cannot provide information about the target genes. So MPRA tells you whether a sequence functions as an enhancer or whether variants affect enhancer activity, but it cannot tell you which gene is affected. And because enhancer could regulate genes over long genomic distance, so the closest genes sometimes not the target genes. And to determine target genes, we need to integrate with other technique. So in summary, MPRA is a great tool to narrowing down hundreds of thousands of variants to a small subset of a few hundred variants that has a functional impact on gene regulation. But to fully dissect the mechanism of genetic mutation, it requires integration of information from many different techniques. And performing MPRA is just one step in this process. So that's it for my presentation. Happy to take questions.
Wonderful talk. Thank you. Looks like we've got one on the side over there. Thank you. Very interesting. You mentioned as a pro for the lentiviral approach that you have the chromosomal context, which is undoubtedly true. But I'm also wondering whether this might also be a con, because you basically have random integration, and aren't you worried then about positional effects like silencing or activation depending on where you land in the genome?
Yes, so there will be positional effects. And one way that we address this is to avoid the silencing effect, that the lenti integrate into a silencing region. We include anti-repressor elements in our reporter construct. So this might counter the silencing effect due to integrating into a silencing region. And another thing is we usually integrate the same construct into multiple different locations and then take the average results. That's another way that we can normalize out these positional effects. So the idea is that if we just integrate a lot of places and take the average, then we will normal the bias introduced by these positional effects.
Hi, great talk. I have a question that when you're fusing your different variants with different barcodes, how do you identify which barcode is being fused to which variant, since will be randomly done during your PCR reaction? Is it the long-read sequencing approach you use for it? And if so, then what is the maximum length that can be read in that case?
Yeah, so in the association sequencing, we usually do pair and reads. So we usually have a candidate regulatory sequence of 300 base pair. So in the association sequencing, the pair and reads will sequence the 300 base pair candidate sequence. And then for the index reads, we'll sequence the 15 base pair barcodes. If you have longer sequences that you might consider other type of sequencing, like using this nanopore or SmartSeq that allows you doing some long-range sequencing.
I see. Okay, thanks.
Hi, thank you very much. My question is also related with barcode. So you mentioned you aim for average 50 barcodes for each variant. Can you comment in the real final library, what's the average barcode, or how variable this barcode number would correlate with each variant? And also, since each variant has been corresponded to multiple barcodes, then how do you, are you just the average of the barcodes to represent each enhancer, or what method you would do there?
Yeah, so we usually average out all the barcodes associated with a particular sequence. And the idea is definitely having more barcodes will give us more robust results, but also having more barcodes increase the library complexity. That's another thing to consider. So based on our experience, if you only want to test whether a sequence has enhancer function, 50 barcodes per sequence is usually enough. But if you want to study the allelic effects, which is usually has a smaller effect size, so we may want to increase the number of barcodes to have robust result. So in these times, we may having 100 or 200 barcodes per sequence.
Hi, thank you so much for your talk. So you had said that practically you found that the library size that you sort of need to limit this sort of assay to is about 100,000 variants. And then I also noticed that you said that you use 10 to 15 base pair barcodes. So I guess my question is, one, why do you think, or what did you see to make the hypothesis that you shouldn't use library sizes greater than 100,000? And could you think about using longer barcodes or different types of barcodes to be able to increase that potential library size?
Yeah, so I think the limitation for the size of the library is actually limited more by the number of cells and the amount of lentivirus you will need to use. I think the 15 base pair barcodes will give you enough coverage to a lot of sequence. But the thing is, when you have a lot of variants to test in a single library, you need use tons of cells and make tons of lentivirus. And then beyond some points, it's just like this number is almost impractical. And based on our experience, the data quality decreases as you testing a larger and larger library.
And with your transient expression system, is it also 100,000, let's say, size cutoff?
No, this number is only for lenti MPRA. For transient transfection, I think it allows a much higher copy number per cell, so it can definitely allow a much larger library.
Thanks.
The cis-regulatory sequences that you assay are pretty long, 300 to 900. How do you construct those?
How do I what?
How do you construct them?
So yeah, we do the oligo synthesis. Like we synthesize the DNA oligo, just like we synthesize DNA primers, but we just synthesize a pool of DNA oligos. This is the latest length that we could achieve, and I think we synthesized 900 from Twist. I'm not sure if they have published this yet, but I think there's some preliminary testing in our lab. But it's definitely more expensive than synthesized 300 base pair.
Hi, I'm curious. So you've talked a lot about like almost every technical point. You're like, and by the way, this varies greatly by cell type. And what sort of types of cellular systems have you been able to use this? And do you ever sort of compare across different cell types to see what the context-specific effects are in different cell types? And how do you like do that differential enrichment analysis?
Yeah, so we've been testing the type of cells. It depends on the question you want to answer. Like for my personal experience, I've been studying a lot of the gene regulation in the brain development. So the cells that I've been used are excitatory neuron differentiate from iPSC, as well as primary cortical cells dissociate from the primary cortex tissue, and also cerebral organelles differentiate from iPSC. And you can also use some neuronal cell lines. So it's very important to select the right cell type because the gene regulation is very cell type specific. So you want to select the right cell type. And then answer your second question, comparison between different cell types. So one recent study is we compared the primary cortical cells with the iPSC-derived cerebral organoids. And then we found that the MPRA results are very comparable between the two cell types. And then based on our previous experiences, is that due to the permissive nature of MPRA technique, so it's usually that you likely to see very similar results between different contexts. But it's also very important to identify the small subset of sequences that do behave differently in different cellular contexts.
Thank you for your talk. I just wonder, maybe I misinterpreted it, but you mentioned that you took 17,000 variants and from whole genome association studies, but only few of them were true hits in your assay. Is that right?
Sorry, could you repeat the question?
So you took a lot of variants from whole genome association studies. You've done your assay, but only very few of them were real hits. Yeah. Is it surprising?
Yeah, so this is not a surprise. So actually, there are other MPRA studies published recently, and I think there's a consensus that only a small portion, like less than 10%, usually less than 5% of the variant tested in MPRA actually having an effect. So this is this observation has been replicated in other labs. And then Another thing for this is that for MPRA, it identifies variants that affects enhancer activity, but the variant could have other effects, such as affecting the post-transcriptional regulation or the splicing. These effects will not be captured in an MPRA. So MPRA will have false negatives. Another thing is that in our experiments, we perform MPRA in book tissue. So it's a mixture of different cell types. And we found many of these variants located in cell type specific cis-regulatory elements. So if we're performing MPRA in purified cell type, we may better identify the variant that having effects only in a particular cell type.
Thank you. I was wondering if you could talk a little bit about how you QC these experiments. So, like, what do you look for after sequencing to make sure that everything went well?
Yeah, so the QC matrix is similar to the prime editing base editing screen. So one thing is looking at the replicate consistency. We do pairwise correlation, calculate the pairwise correlation coefficient between replicates. So we're usually aiming for looking at correlation above 0.9. And another thing is looking at the controls. We include positive and negative controls. So we expect them to behave in the way we expect. So positive sequence have a high activity and negative sequence have a low activity. So that's the two commonly matrix we use to QC the results.
Have you tried using promoters that have really high activity and really low activity to look kind of both ends of the dynamic range, like variants that suppress transcription, or are you just looking for on signals?
Yeah, so, well, that's a good question. So currently, the MPRA construct we use now include a minimal promoter, so it has a very low basal activity, and this is ideal to identify enhancer activity, like increased activity. But if you want to study silencing effects or the silencer, it's actually better to use a strong promoter, so you can looking at the repressing effects.
All right, well, thanks for a great talk, and let's all thank you.